Hi, this is the Infection and Response knowledge test. Okay, now what is a communicable disease with 3 examples? So what communicable means is it means it can be transmitted to somebody else, right, by some sort of contact of sort. So basically a communicable disease is a disease transmitted from person to person. Okay. Right, now 3 examples, right, there's many examples that you can actually have, right, so I'll go for measles, I'll go for HIV, AIDS, and I'll go for typhoid. Okay, so they are then 3 diseases that can be spread quite easily from person to person. Question number 2, what is a non-communicable disease? So if a communicable disease is one that can be spread, a non-communicable disease, right, is one that cannot be transferred. Okay, so it can't be shifted from one person to another, right? It's not normally a disease either, it's kind of like a condition. First example is diabetes. Okay, obviously you can be born with diabetes, type 1 diabetes, or you can get type 2 diabetes that can develop in later life, often due to your lifestyle. Cancer. Okay, cancer cannot be transmitted, whatever sort of cancer it is. Right, another one is Alzheimer's. Now, I generally hope I've spelt that right. Okay, which again, something that develops usually later on in life, but can be earlier. Question number 3. Name 5 factors that can significantly affect health. Right, now, there's loads, right? So what I'm just going to do is I'm just going to go through some of the most obvious ones. Smoking, right? To be honest, if you smoke, then, you know, you're daft. The next one is obesity. Okay, that is then when somebody is then overweight, right, considerably overweight, right, due to their diet, due to lack of exercise. The third one is alcohol. Okay, if you drink too much, right, then it will significantly affect your health. Carcinogens, right, is exposure to things that can cause cancer. All right, so maybe things like radiation. Carcinogens, that's an S on the end there. And the last one that I've got there is if you smoke or drink when pregnant. All right, so that's not necessarily affecting you, even though we have talked about these already before, but if you smoke or take in alcohol when you're pregnant, then that is a massive, great big no-no. Okay, it's going to significantly affect the baby. Question number 4, what is a malignant tumour? Right, so a malignant tumour, right, and a benign tumour is something that will not spread, right, and to a certain extent it isn't kind of danger of life, where a malignant tumour is something that is cancerous and can spread. Okay, so what a benign tumour tends to be, it tends to be within sort of a bag, a package, it sort of tends to be within something, right? A malignant tumour is where it's starting to grow roots, it's starting to escape out of whatever package it could be, so that then what it can do, it then can spread. Question number 5, list 5 ways germs can be spread. Okay, so number 1 is direct contact. Okay, so if we got direct contact, that could well be touch. Right, so if I got germs on my hand and what I do is I then shake hands with somebody, right, then that's direct contact. I could pass those germs on, they could then touch their face and it gets taken in by them. We could have indirect contact, right, so that could be like a door handle, right? So somebody's got germs or pathogens on their hand, they touch a door handle, they then go into the room, I then come along and I do exactly the same thing, right? Then I would then have them on my hand. Another one is a vector, right? And that can be by something like a mosquito, right? So a vector is something that carries it. And a mosquito is the perfect example, where what that will do then is that will then transmit malaria, right? And they're usually a protist. It can also spread through food, and it can also spread through water, right? So you can quite easily have germs on food and you can eat it, like salmonella in chicken, salmonella in eggs, salmonella in other types of food, and water, where you might get a waterborne disease that can also be. Question number 6. What are the 4 main groups of pathogens? Oh, look at that, I didn't do I did a space there, which didn't make that look very good. Now, these first letters annoy me, so I'm just going to write down virus. I'm going to write down bacteria. I'm going to write down protist. And I'm going to write down fungi. Okay, right. Now, examples. So we've got virus, bacteria, protist, and we've got fungi. Good examples of a virus are flu and HIV. Didn't do H very well there. So flu, right, and HIV. Bacteria, there's loads and loads of different examples. We've got TB, tuberculosis, and I'm not going to fit this one in, salmonella. Salmonella, right, they're bacteria. Now, the big difference between really virus and bacteria is the fact that bacteria, they can be treated with antibiotics, where viruses can't. Viruses will then have some sort of antiviral drug which reduces the kind of effectiveness of them. Protist, dysentery, but then the big one is malaria. And in a later video, what we'll do is we'll go through malaria in a bit of detail. Fungi, what we've got here is we've got athlete's foot, and we've got rose black spot. Okay, so what I've done on fungi is I've put in an animal one and a plant one. So an animal one is kind of us, right? That's kind of when you get athlete's foot, is where it's like a white powdery stuff in between your toes. Rose black spot is obviously rose and there's black spots all over it, and that's a fungi. Now, special features. With a virus is very simple and very small, and what it is DNA surrounded by a coat, right? It's not like a coat like you might wear, right? It's kind of almost just like a membrane around the outside of it, right? Sometimes they look really weird. They look like almost like alien-like. Bacteria is a prokaryote, and the key thing for a prokaryote is there are no membrane-bound organelles, right? And what that means is it's got no nucleus. Oh, that D is terrible. P, protist, is they are single-celled. Okay, so what they are is just one individual cell. Fungi can be single or multi-cells, right? So some fungi can be incredibly small, some fungi can be absolutely massive, and it kind of just depends on which one we're talking about. Question number 7. What are the 4 main ways your body prevents entry of pathogens? Right. So what people always say on this one is they always say, oh, you've got white blood cells, right? But read the question here, 4-way prevent entry. All right, so I'll do it in purple, right? Prevent entry is, number 1, is your nose. And what that does, it has hairs and it has mucus. So the hairs and the mucus catch any sort of pathogens that go in through your nose, and eventually what they'll do is they'll come out. Number 2 is your trachea, okay, so your windpipe. So when you breathe in, that has also got mucus. And so your pathogens go down your throat, they catch in the mucus, and then what you'll do is end up coughing it out. Number 3, stomach acid. Okay, could be pH 1, pH 2, right? Very, very strong acid, right? And literally, if it goes into your stomach, right, the main job of the stomach acid is to actually sterilize the food, which is to kill the bacteria. Number 4, the big one, is your skin. And what that is, that's a protective layer, right? And the skin for a bacteria is difficult to penetrate. Okay, so if you do get a cut, your body responds very rapidly to clot it, right? Because your body doesn't want to allow any form of bacterial pathogens to get in. Question number 8. What are the 2 types of white blood cells and what are their roles? Again, I've got the first letters there. We've got phagocytes, right? And what they do is their role, which sounds very similar to phagocytes, is phagocytosis, which basically means they engulf and then they digest. Right, so they go along a bit like a Pac-Man, right? So what they do is they go along like that, they find the bacteria and they go, oh, right, and they'll eat it, or not eat it, they'll engulf it, and then they digest it when it's in. The other one is lymphocytes. Okay, lymphocytes. They've got 2 jobs. Job number 1 is they produce antibodies. And what antibodies do are antibodies attached to the pathogen, right, that is within the body. And they either kill them directly or they kind of clump them together and they kind of enable the phagocytes to find them easier. And the second job of a lymphocyte is antitoxins. Okay, so that's what they do, right? So what a pathogen will do, it'll produce some sort of toxin, some sort of chemical that makes you feel ill, right? And what the lymphocytes do, they produce an antitoxin that then stops you feeling quite as ill as you were before. Question number 9. Name 3 drugs that were extracted from plants or microorganisms. Drug number 1 is digitalis. Okay, so digitalis is a drug that is used to do with the heart, and it's found in the foxglove, right, which is a beautiful tall plant. You know, it can be a metre high and it's got fantastic flowers, but it's got digitalis in it. Number 2, aspirin. Okay, so aspirin, right, what it tends to be used for, it tends to be used for pain. It can also be used for thinning blood. Okay, and that was found in the willow tree. I have no idea how they actually found out that the bark on a willow tree has got aspirin in it. And number 3 is penicillin. Okay, oh God, I can't write at all on this. Penicillin is an antibiotic, okay, and it's basically from a mold. Question number 10, right, and then we're talking about drugs, right, so what are new drugs tested for and what do the words mean? So first one is, with an F, efficacy, right, efficacy is making sure it works. All right, so you create a drug that is designed to get rid of cancer, right? But then it doesn't do it. Then what you've got is the efficacy is not good enough. So efficacy is making sure the actual drug works. The next one is toxicity. Okay. Now that then means you've got to test to see if it's toxic, right? And you've got to make sure it isn't actually going to be poisonous to the person, right? So you find a cure for the common cold, works brilliantly, efficacy is fantastic, but it kills you, right? Then obviously that isn't a particularly good idea. And the last one is dose. So what people will do is they'll test for the amount of dose that's required. So with paracetamol, often an adult will take 2 tablets at a time. And what they'll have done, they'll have gone through the dose stages to find the suitable amount. Right, number 11, right, what are the 4 stages of clinical trials? So we're talking here about clinical, which is then when we're talking about with humans, right? Preclinical trials is when you're talking about doing drug tests on cells or small animals like rats or guinea pigs, or large animals like dogs, pigs or monkeys. This is then the 4 stages of the clinical trials. Stage number 1 is healthy volunteers. Okay, so healthy volunteers. So any of us, in theory, could volunteer to do a drug trial. And the whole point of it is to see if the drug is safe and to see what side effects there are. All right, so you've got a normal, 23-year-old bloke, 23-year-old woman going in, and what they'll do is they'll take a certain amount of the drug just to see if it's literally safe to take and what the side effects could possibly be. If it goes through that and everything's fine with that, then what they'll do is they will then do the actual drug trial on patients, but a very small amount. And what they're going to do this time is see if it works. Right, so then they're looking at the efficacy. Right, so you've got a drug that is designed for get rid of a certain type of cancer. You give them the drug. If it gets rid of that certain type of cancer, has an effect on that certain type of cancer, stage 2 is good. Stage 3, they will then do it on more patients. And when they're doing it on more patients, they found out that it actually does its job. They found out that the side effects aren't too bad and it's relatively safe. This then is to find the optimum dose. Okay, so there's no point in giving too much, right? There's no point in giving too little. What you want to do is you find the best dose to cure the problem. Stage 4 is then a double-blind trial. Okay, that is then when the patient and the doctor don't know who's got the drug. So let's say they've got 10 patients that are actually part of the double-blind trial. Half of them will get the drug, right, and the other half will get the placebo. All right, but what they do is they do it so that the patient and the doctor, so both of these people don't know if they're getting the drug, right, because there's kind of like an effect that somebody who thinks they're getting a drug might get better. Right, the other half get a placebo, which is exactly the same thing but without the active ingredient. Plants, let's go for green on plants, that'd be a good colour. Why or how do plants protect themselves? They protect themselves in a physical way. For example, a waxy layer. They protect themselves in a mechanical way. For example, thorns. And they also protect themselves in a chemical way, which is things like antibacterial or against toxins, or they produce